In this video, we present Coral, a web-based visual analysis tool for creating and characterizing cohorts. Coral is joint work between the Visual Data Science Lab at the Johannes Kepler University Linz and Boehringer Ingelheim. Coral follows an overview detail approach in which two components are shown in a horizontal split-screen setup. The cohort evolution view shows a graph to track the creation and evolution of cohorts. The action view allows users to further characterize cohorts or to derive new cohorts by applying various operations. It is divided into three areas: the input area that lists the selected cohorts from the cohort evolution view, the operation area with all available operations to create and characterize the selected cohorts, and the optional output area that lists the newly created cohorts. The input area and the output area both use a tabular layout that shows cohorts and the attribute distributions. Coral is preloaded with data from the AACR Project GENIE, with data from TCGA and CCLE, and with two depletion screen datasets. We demonstrate the use and utility of our tool by reproducing findings from a recently published article about KRAS G12C somatic mutations by Nazar and his co-authors. We start the analysis by loading the GENIE dataset. We then want to create two subcohorts containing non-small cell lung cancer and colorectal cancer samples. To do so, we first filter the GENIE cohort using the tumor types that comprise the major subtypes of non-small cell lung cancer. Next, we filter the GENIE cohort using the colorectal cancer tumor types. To get an initial impression of the demographics, we select both tumor type cohorts and visualize the distribution of the numerical attribute age. We see that on average, non-small cell lung cancer patients seem to be older compared to the colorectal cancer patients in the GENIE dataset. To confirm that this difference is statistically significant, we switch to the compare operation. From the comparison table, we see that the p-value is less than 1,000th part. Additionally, we investigate potential differences regarding the gender distribution and see that the non-small cell lung cancer cohort has a significantly higher proportion of female patients. Subsequently, we want to check potential race and gender differences in the data with respect to the KRAS G12C mutation frequency. For this analysis, we focus on the non-small cell lung cancer patients. We start with a split operation using the categorical attribute race. We then create cohorts for the three largest populations in the dataset, namely white, Asian, and black. Next, we generate 6 new cohorts via the split operation on the categorical attribute gender. We visualize the frequency of the different KRAS mutations for each cohort. We can see that KRAS G12C mutations in non-small cell lung cancer are less frequent in Asian women compared to Asian men, but more frequent in black and white women compared to black and white men. Furthermore, the prevalence of this mutation is overall lower for Asian compared to black and white. These observations agree closely with the findings of Nazar and his co-authors. We generate new cohorts for samples with the KRAS G12C mutation to further investigate them using the prevalence operation. Here we can look at the prevalence of the cohorts visually and get additional information about the confidence intervals. We can dynamically change the reference cohorts that are used to compute the prevalence estimates. This short demo use case showed the utility of Coral and highlighted how easily even complex scientific questions can be investigated. You can try Coral yourself at coral.kaleidoapp.org. Thank you for watching our video.